In this lecture, we're going to continue our investigation of the chemical nature of many of the particles that we need to consider when studying cell physiology. And we start with acids and bases. And an acid refers to a substance or a molecule that releases protons when dissolved in water or in physiological solution. And so some examples of acids that we will address throughout this course, and we'll talk about that here today, is hydrochloric acid, which is a molecule produced by the cells inside the human stomach, and lactic acid, which is produced by muscle cells. And we'll use that as an example here in the next few slides. The opposite to an acid is a base, and a base is a substance that can neutralize the effects of an acid, and specifically these are molecules that provide what's called the hydroxide anion, and that's an oxygen atom bound to a hydrogen atom that has a net negative charge opposite to that of a proton that has a net positive charge when that is dissolved in water. And so some examples include lye, caustic soda, and sodium bicarbonate. And so if people consume an antacid, a tablet that goes by some of the brand names of Tums or Rolaids, the person is ingesting sodium bicarbonate in an attempt to neutralize excessive acid produced by the stomach. Now in physiology, acids are the one type of molecule that we will discuss to some extent here. And so we start with the question of whether acids are good or bad from a physiological perspective. And so the answer to this question depends. Now, to illustrate this answer, we use the example of the 400-meter race in track and field. And so this is one lap of a track that runners race at their maximum speed. And so this is a very difficult race and requires a high level of muscle contraction. And so in order to fuel this physical activity, the muscle cell must spend ATP, necessitating the muscle cell to resynthesize ATP at the same rate that the muscle cell spends it. Now, in order to make that ATP, a fuel has to be combusted. And one of the premium fuels that's used in this high-intensity exercise, this high-intensity race, is a sugar that we call glucose. So inside a skeletal muscle cell, we know that to produce force, it costs money. And that money, in the form of what the muscle cell will recognize, is called adenosine triphosphate. Now, in order to match the ATP that is broken up to harness its stored chemical energy to drive muscle force, the muscle must respond immediately, and to make dollar for dollar, ATP for ATP, that molecule at the same rate that it is spent. And the metabolic pathway inside the muscle cell is called glycolysis. And so glycolysis is the metabolic pathway in which a cell uses to quote-unquote burn glucose, break up glucose. And in the process of breaking up glucose, ATP is produced. Now, when a muscle cell relies on burning glucose at a very high rate, one of the byproducts that is produced is this compound called lactic acid. So lactic acid derives from a glucose molecule that is being metabolized at a very fast rate inside the cell. So here you see the chemical formula for lactic acid, and this is an illustration of the chemical structure of lactic acid here on the left-hand side. And so a couple of key atoms here that I'm going to be focusing on is we have a carbon atom, we have it bound to an oxygen atom, and we have another oxygen atom, and out here is a hydrogen atom. Now, this by definition, according to our previous slide, is an acid. And by definition, an acid is a molecule when dissolved in solution. In this case, it's the cytoplasm of the muscle cell will unload, will release a proton into that solution. So when lactic acid loses a proton, this oxygen atom right here, so the red indicates oxygen atoms, this oxygen atom will have a single electron attached to it. Now recall, hydrogen, it's one proton and one electron, but if we lose that proton, what's left is one electron, and that's what we see here. So the overall molecule now carries a net negative electrical charge. And we call that the conjugate base of the acid, of any acid. So any acid, once it loses its proton, what's left over is what we call the conjugate base. And for this particular molecule, the conjugate base of lactic acid is called lactate, and it's pretty small here, but you see the superscript of a negative sign indicating that lactate has a net negative charge. And what's left over is that hydrogen ion, or a proton, that enters solution, and it's the proton that is responsible for increasing the acidity level of that cell. Now, with respect to lactic acid inside the muscle, this does pose some particular problems. So, in large enough concentrations, if lactic acid accumulates and once it's produced, it immediately spontaneously dissociates into lactate and the proton, that will contribute to muscle fatigue. So the person will not be able to run the 400-meter race at full speed without eventually slowing down. And the lactic acid, particularly the proton, is the element that is responsible for that burning sensation that people experience, that pain, discomfort when engaged in high intensity exercise. So what we've seen so far is that acids appear to be bad for physiology, but there are instances where acids are actually considered good, and that is found inside the digestive system. And so when humans consume dietary protein, that protein must be broken up, must be digested, so that we can absorb the individual amino acids from the digestive system into the cardiovascular system. The whole protein in the food is too large to move from one system to the other, so in order to gain a physiological benefit of that dietary protein, it must be broken up into its constituent amino acids, which can then be absorbed. And so some food sources of protein include milk, eggs, beans, tofu. Protein is found in a wide range of different foods, but to different extents. So protein is a complex, stable molecule, and it must be what's called denatured from a dietary perspective. The term denature indicates that we're taking a molecule in its normal state and we're breaking it up. And that's what happens inside the stomach. Now, how does the stomach break up that dietary protein for our physiological benefit? Well, the cells of the stomach secretes a compound called hydrochloric acid, one hydrogen and a chlorine atom. Now, it's an acid, and once this acid is produced, it does the same thing as other acids. It will unload a proton. Now, that proton comes from the hydrogen, so when this hydrogen loses a proton, all that's left behind is a single electron that the chlorine atom possesses. And so the chlorine atom, which is normally electrically neutral, now has an extra electron. And at that point, we no longer call it chlorine, we call it chloride. Now the reason we lose the H symbol here, because the H symbol is defined not by the electron, but by the proton. So if HCl loses a proton, the H is lost, but the negative sign stays behind. And so we now say that chlorine has been ionized to chloride. And it's this acidic environment caused by the protons which creates a much more unstable dietary protein that starts to fall apart. And so the pH scale is a measurement of the relative acidity or alkalinity of various substances. And so the major factor that dictates the pH level of a particular molecule or of a solution or of an acid is determined by the proton concentration. So when a molecule releases a proton, that proton will bind to H2O molecules in that solution. And so when an H2O picks up a proton, remember, the proton is the hydrogen atom that has been ionized. And so we see an H2O, because it picks up a proton, which is hydrogen, the H2O becomes an H3, but without the additional electron, H2O becomes H3O with a net positive charge. This compound is now referred to as the hydronium ion. And as that concentration goes up, the pH value actually gets to a lower value because it's defined as the negative common log of the hydronium ion concentration. And the scale on the right-hand side, we see different substances, different solutions, and their relative pH. So if we take a look at battery acid, sulfuric acid, these have very low pH scores, indicating these are very acidic. On the opposite end, items such as bleach, drain cleaner are not acidic, are very alkaline. So these have a very high pH value, indicating the proton concentration is very, very low. In terms of human physiology and pH, we see the neutral level of score of 7, which is halfway through the full range of the scale from 0 to 14, that human physiology likes to have a nice neutral pH of around 7, slightly higher, slightly more alkaline. If we go with 7, that's perfectly neutral. We see that human blood, the interior environment of skeletal muscle, is also slightly alkaline, moves a little bit away from the acidic side, but it's still fairly neutral. The human stomach is very much on the low end of the pH scale, which is to say it's on the high end of the acidic scale at about 1.5, somewhere between vinegar and sulfuric acid. And so normally in other parts of the body that would be damaging to cells. However, inside the stomach, hydrochloric acid is very effective at driving a breakdown of dietary protein. So this ends this part of our lecture on cell physiology.